All right, so I think what we'll do is we'll start. First of all, I'll ask again for all the latecomers. Do you guys have any questions from last class about synaptic transmission, the basics? We ended at NPQ, which are the properties of synaptic transmission. But I'll start with asking you a few questions, okay? And let's see if your concepts are right or not. Let's see. So I told you about the different types of receptors, and all these receptors can have kind of two, they can be divided into two categories, mainly one which is inotropic, which participates in the synaptic transmission. And then we have the other one, which is like a metabotropic receptor, which is more of a neuromodulator, right? Or it activates a GPCR-based signaling. And so we have three categories here for acetylcholine. You guys now know which is the inotropic one. Nicotinic, you can read it. Okay, it's not too small of a font. And then muscarinic is a GPCR one. And then for glutamate, you have AMPA-kainate. We have metabotropic glutamate receptors. And today's lecture, we'll focus on the NMDA receptor glutamate and how it plays role in changing the properties of synapses. Again, GABA also has this inotropic and metabotropic receptors. So GABA A is inotropic, GABA B is metabotropic. Now, last class, I described to you guys about IV curve. Do you guys remember what that is? Current voltage relationship. How does can someone draw the current voltage relationship of a potassium channel? Someone wants to draw here. Are you guys confident? No. Okay, that's good. That's good too. What just generally it's a linear IV curve, right? And then what about sodium? Yeah, so it basically exactly like that. And what is the voltage where it is crossing? So let me just draw it quickly, right? So you have an IV curve and you have nothing, nothing, nothing. And then suddenly you have, this is your potassium, right? And then for sodium, you have nothing, nothing, nothing. And then it goes up and then it goes like this, right? And so what is this? Where it crosses. So here the currents, this is current, this is voltage, this is positive, negative, negative, positive, right? So this current is negative, and then here it goes and changes to positive. So this is your reversal potential. Exactly. This is where the current direction reverses. Okay, cool. So now if we think about the IV curve of AMPA receptor, can anyone draw the IV curve? Think about how an IV curve of an AMPA receptor look like. So what is one main difference between an AMPA receptor and a sodium channel? Can anyone tell the difference? Sodium channels are voltage gated. Okay. Okay, so that's exactly right. So sodium channels are voltage gated, AMPA channels are ligand gated, right? All these receptors that I talk about, these are ligand gated ion channels. Now, how would an IV curve of AMPA receptor look like? Does it depend on voltage? No. After a point of time, it will saturate, meaning? So imagine all the. That is true, but we are not talking about the temporal component, right? We are talking about the IV curve, meaning that at different voltages, how would the current look like? It's independent of that, okay, but it is still dependent on what? It is still dependent on the ionic, the concentration gradient of the ions, right? It is still permeable to sodium and potassium, and it's still going to be, but it is independent of voltage. So whatever, so you have a particular voltage, you'll have a current. If you're far away from the reversal potential, you have a lot more current. If you are closer to the reversal potential, you have lot less current flowing through the AMPA receptors, right? And that's exactly what you'll see here. So you'll see an AMPA receptor curve, something like this. So did not change here. It changed, right? So this is what an AMPA receptor IV curves look like. Where you have a lot more current when you're far away from the reversal. The reversal potential is here. After this, the direction of the current changes. So if you're far away, that means your concentration gradient or ionic gradient is a lot higher. And so the driving force will be a lot stronger. And if you're going closer and closer and closer, your driving force is reducing, right? Now, there are many cool properties. I'm not going to go into detail of the AMPA receptors, but AMPA receptors are generally heterotetrameric in structure. So they're not really existing as one AMPA receptor subunit. They are usually in a tetrameric structure. They have different subunits. We have GluA1 to GluA4. And one of them, which is GluA2, is the one that provides calcium impermeability. If that doesn't exist in your heterotetrameric structure, the AMPA receptors will become calcium permeable. Okay? So GluA2 is the one that provides calcium impermeability, and that's one of the subunit that predominantly exists in all AMPA receptor structures. Okay? And how does it get its calcium impermeability is also very interesting phenomena. It's due to something called as a QR editing, RNA editing. And so you read about that and you tell me. Okay? I'm not going to go into detail of that. And also, if you guys can tell me, this is a bonus question. If you guys can tell me, what would an IV plot of AMPA receptors that lack GluA2 look like? So that is not a very easy thing to, you need to know some properties of GluA2 structure for that. Cool? So 2 questions. All right. So the third thing that I want to show again is the NMDA receptor IV plot. Okay? Since we are talking about glutamate receptors. So NMDA receptors IV plot looks something like this. And do you guys know why? It's also like kind of non-linear. So there is some voltage dependence. It's not a voltage gated ion channel, right? It's still a ligand gated ion channel, but there is some voltage dependence on this associated with this channel. And what is that? Do you guys know? Yeah. So what happens is at hyperpolarized conditions during normal synaptic transmission, NMDA receptors don't participate because there is a magnesium block that even if the glutamate comes in binds, it's not going to get activated. Only when you depolarize enough, you will have activation of NMDA receptors. You keep depolarizing, you'll have more activation of NMDA receptors because there is magnesium block that gets released. Okay, and then again the reversal potential will be close to 0. Acha, one thing, why is the reversal potential close to 0 for AMPA and NMDA? What does reversal potential? Why is it 0? What does it mean? It's not, for sodium it was 55, for potassium it was -90. Why 0 here? Can anyone think of? I gave you the answer literally. Yeah, exactly. So it is permeable for both sodium and potassium. So that's why the reversal potential is somewhere in between, which is like around 0. Okay, so it's not specifically permeable for sodium or potassium. Okay? None of these channels. All right, so this is the property of NMDA receptors that makes it the receptor or the ion channel for or participating in learning or participating in synaptic plasticity. And so we'll learn about how it participates in that. And it's very important that it doesn't play a role in the basal transmission or day-to-day communication. It only gets activated when something important is happening. Okay? Yeah. Receptor. Sorry. So this is a typo. So it should be NMDA receptor. Good catch. So again, NMDA receptors act as heterotetramers. So you have 4 subunits of NMDA receptors as well. And if you'll go into the detail of NMDA receptor, we can have like several lectures on these subunits and what's the function of these subunits altogether. Like they perform very specific either anchoring role or structural role where they can really maintain the signaling cascades in an active form. Okay. Anyway, so let's start the synaptic plasticity lecture. When you depolarize, and you have some changes in the membrane potential in the positive direction, you have magnesium relief. There's a conformational change. C graph, let's see. So this is the extracellular magnesium concentration, and this is the peak amplitude of EPSC. So if you have lower concentration, you'll have much higher peak of EPSCs at -60. So -60 is somewhere here. If you don't have magnesium, you will have, even at -60, it will almost act like an AMPA receptor, right? So the glutamate binds, no magnesium, so there's no blockage. So you'll have much higher peak. But as you increase your magnesium concentration, you'll start to see that at -60, you'll start to see a dip in the current. But they are only recording. So this is again a voltage clamp. You're clamping the cell at -60, and then just recording it at different extracellular concentrations of magnesium. Cool. All right. So let's start with synaptic plasticity. I don't know why it's still showing winter. This is summer lecture in June 2025. All right. I think I changed it. So let's go into the history of synaptic plasticity. The concept of neuroplasticity is something that psychologists have thought for like centuries. I think the concept that the brain functions are not fixed and they keep changing across one's lifespan has been systematically characterized or like thought about by William James. Should check out his book, Principles of Psychology. He's done a great job to like introduce the concept, and we are still testing some of them today. One of the when you talk about plasticity, one of the things that you guys should know, and this sentence that you should know, is this Hebb's postulate, where again, he was a Polish psychologist, and he wrote this book, Organization of Behavior. In that, he talked about how now during this time, we already know anatomically about the neurons being the functional unit of the brain, and they are contiguous and not continuous. And so people have started to work on those theories. And this guy now talks about how the neurons learn. So he says when the neurons get activated together, when they fire together, they wire together. And what he meant by that is, so I'll just read it out. When an axon of cell A is near enough to activate cell B repeatedly or persistently, such that the information is coming again and again and again to the cell B, there is some metabolic change that takes place in one of both cells, such that A's efficiency as one of cells firing B is increased. And he could not be closer to the truth, right? So this is 1948, 1949, and we are really following some of his Hebbian principles of there is actually some change, some metabolic change or activation of some signaling cascades that happen that leads to synaptic plasticity. So I'll basically describe the evidence of this statement, yeah, in this year. So before I go into this, then people started in 1940s, we had no idea which brain region was actually important for memory formation, and there are different types of memory. Psychologists knew that you can categorize memories into different forms, one where you consciously need to think about, which are declarative memories, one where you're not consciously thinking about, like writing or bicycling or reading. Those are non-declarative memories, right? So they were categorizing them, but in the brain structure wise, they had very little idea of which brain structure was responsible for what. And then this example of Henry Molaison comes. You guys know about Henry Molaison? So in this patient, he was an epileptic patient, and he had some bike accident when he was 13, 14-year-old. And they tried a lot of anticonvulsants. And by, I think, age 26, 27, they had to remove bilaterally the medial temporal lobe of that patient. When they removed, they found that this guy, he knew everything. He knew who he was, where he was born, but he was not able to make new memories. And he had this problem of retrograde amnesia, right? And he's one of the most studied patients, where they tried to do a lot of different experiments. Some of them were actually funny. So they would come, they would introduce themselves, and they would just slap this guy, right? And they would go back. And 15 minutes later, they will come again, and this guy will not remember that these guys have actually slapped him, right? They would do that for 2 days. And after 2 days, he would like associate the pain with these guys, but he would still not recall, he would not be able to recall why he got that pain, right? And so they did a lot of, so Brenda Milner was one of the scientists who studied this patient for, I think, decades, if I remember. But that was a time when we kind of discovered that, okay, hippocampus is important. If you remove it, it does have, like, it is the focal focus of during the epilepsy. It does start there, but if you remove it, it's actually, you know, causing something even worse. And so scientists then used animal models to study hippocampus, and they wanted to test this Hebb's postulate. So one of the first things they did, and this is the evidence of, first evidence of changes in the synaptic strength. Okay? This is anesthetized rabbit, Tim Bliss and Lomo. They put in a stimulation electrode. So you guys know about the structure of hippocampus? I'll just tell you briefly. So hippocampus, again, if you see in the brain, these are like these horns, ram's horns in the medial side. And if you make a coronal section like this and open it up, it looks like 2C. This is something that you might have seen. This is the dentate gyrus region. This is CA3, and this is CA1. Okay? And you get inputs CA1 from entorhinal cortex, or you also can get from entorhinal cortex different layers to the dentate gyrus. Okay? Now, what these guys did, you see here, this is exactly what they are showing in the slice form, but this was in an in vivo rabbit, where they put an electrical stimulus into the entorhinal cortex inputs coming onto the DG. Okay? And then they were recording from the DG. So what they are doing is they're just stimulating the axons, presynaptic axons, and recording from the somatic region. Now, even here, they had two configurations. One, so hippocampus is beautiful in the sense that you have the soma, you have the dendrites, you have the axons. So the inputs and the outputs of those hippocampal structures are actually very well segregated. Okay? And here, they were stimulating and they were recording from either the soma region, where you have the soma, where the action potentials are going to form, or they were recording from the dendrite region, where you will see the synaptic activation. So if they would record from the dendrite region, they would get something called as a population EPSP, which is called as excitatory postsynaptic potential. Population meaning from many cells, because they were not recording from inside. And then here, if you record from the soma region, they would get something called as a population spike. So like many cells at the same time getting an action potential. So this is kind of like a way to look at synapses and how do these synapses change if you give patterns of electrical activity. Now, to test the idea that repeated or persistent activation of some synapses to the postsynaptic cell can change, what they did is they gave 100 hertz stimulation at this time. So they recorded some baseline for like maybe 30-40 minutes. Then they gave this 100 hertz stimulation. Initially, they did not see much. Then they gave, after 30 minutes, again, another 100 hertz stimulation. Like this is just repeatedly activating the very fast or repeatedly and very fastly activating the presynaptic axons. They did not see much. But then after the second one, they start to see that only in the inputs or on the set of axons where they were stimulating or giving this stimulation, only there they start to see an increase in the population spike and also increase in the EPSP amplitude. Okay? And this is the other control where they were also recording the responses from this other group of synapse. Like imagine you have one stimulation here and you have like many of these inputs coming in and you have another stimulation here. But only in the ones where they gave this 100 hertz stimulation, they saw an increase in the synaptic strength. And they recorded this for several hours, 6 hours, and the potentiation or this increase in the synaptic strength was consistent. And this then became the model to study plasticity, or like this, or plasticity and the molecular mechanisms that underlie that for the neuroscience field. This was the first evidence that came. Okay. Now, if you just, now we know, like now we have done this experiments for 50, 60 years, and we know that there are different phases of hippocampal LTP. This is also true for other synapses, mossy fiber CA3 synapses, and then CA3 to CA1 synapses. This potentiation happens in all these different synapses. Okay? And if you look into the hippocampal LTP phase, it can be divided into 3 distinct phases where there are different mechanisms happening or different signaling cascades that get activated. Okay? After recording a baseline, you have pathway 1, where you do a high frequency stimulation, and there is an induction phase where you induce, you activate a lot of signaling cascades, and then you have an expression phase where there's a different, we hypothesize, we don't know for sure, but we hypothesize that there's a different set of signaling cascades that stabilize this potentiation. And then after the expression, you have the maintenance phase where now it's stable, but we want to kind of maintain it. So that's the stabilization or the maintenance phase. And if, so you can have some potentiation depending on the type of learning that can just get induced, but it doesn't get stabilized. So it can go down very quickly. Then you can have some plasticity that can be there for some hours, and then some stay for days. Even if you record 4 days later, you can really see the increase in the synaptic strength. And we'll learn about the different mechanisms that could participate in some of these. All right, so let's understand the theory of what do I mean by strength, changing the strength of a synapse. Okay? Yeah, so imagine you have 3 neurons that are coming in and you have a postsynaptic dendrite where all these synapses are made. These are, I'm assuming these are all excitatory in nature. They all start with the same strength. Now you have neuron 2 that keeps getting activated. Persistent firing happens. Okay? So there is some metabolic change such that the synaptic strength increases. Okay? And what does that mean? That means, can you guys think of how do you define a synaptic strength? Here I keep talking about, I talked about changes in the population. So there's an increase in the amplitude of a population EPSP, right? But how would you really define theoretically? That's just a measurement, but how would you really define changes in the synaptic strength? Do synaptic activation lead to firing? What is a synaptic strength? How would you define that? That is a way to change the strength. But how would you define strength? Number of cells? So here, though, we are not even talking about, so this is, imagine 1 cells, 1 axon activating 1 synapse, right? And you want to measure, you want to say that this synapse is stronger than this synapse. You're getting closer. Yeah, so yeah, so you guys are getting closer. So imagine you have activation of 1 synapse that leads to some depolarization. You guys have seen this in Subhashish's lecture. But if a synapse is stronger, you get a much bigger response, right? What is, so the way you define this is that how 1 synapse contributes to the generation of an action potential as compared to the others. If the synapse is stronger, that means it contributes to, or it brings the cell much more closer to the threshold than the other synapses. So that's how we define the synaptic strength. there is the chances, the probability of action potential to get fired increases when the neuron 2 or the synapse 2 becomes stronger. Yes, that's how I'm going to end. That's exactly what we are setting up in this. We can really record now from individual synapses the current responses, and then we can compare the synaptic strength in an absolute term. Okay. So now how do we change the strength of a synapse? You guys know the basic structure. It's actually very simple. It's so simple that I'm actually, surprised. Why do we need thousands of proteins to do this simple phenomena? So the basic structure of a synapse, what are the three properties that we learned last time? What are the different parts of the synapse? What is this? Synaptic vesicles, you have synaptic cleft, you have number of receptors, right? And so what could be different ways of increasing the strength? Okay, good. You increase the number of vesicles and also the probability of increase. It comes in the same category. That's fine. What else? Number of receptors. So now I'm saying that this is already too many receptors in the synapse. So I need to increase. For increasing the number of receptors, what I need to do? You need to increase the area. You increase there should be a change in the spine volume that can incorporate more receptors, right? So this is the second way you can increase the spine size and the receptor number, right? And then the third one is a little bit more dramatic, where you just have more spines. You just have there's something called as a synaptogenesis. You just have new spine for the same presynaptic partner. And this happens a lot more in a younger humans and younger animals. Surface area. So let's see if I can. This is something that we don't know. Why one thing happens in some circumstance than the other? These are the three types of observation that we have made, but we've actually physiologically or behaviorally don't know why this happens. I can tell you one thing. Sorry, you want to? Okay, there's a limitation of the surface area, but why can't we have more? So what happens is there is an actin cytoskeleton that gets polymerized, and you can just polymerize that. Energetically, I know that this is a lot less energy consuming than that. So doing that is a lot more dramatic event, which is energetically more stable. These two. But why can't we maintain this polymerized cytoskeleton? So once you've polymerized it, we can just keep it in a stable condition, no? If there are some cytoskeleton structural proteins that got activated and they did the polymerization, and then we've just maintained it that way. I don't think stabilization wise, they both are kind of similar, because once you make a new spine, you can also have ways to stabilize it. But why you would choose one over the other, I don't know. There could be, again, differences in how you are inducing plasticity, whether a lot of presynaptic information is coming in to lead to the postsynaptic changes, or there are changes that are happening postsynaptically, or a third neuromodulator is coming to increase the spines. So I don't know. The population of readiness, absolutely. That is, again, very good point. Something we don't know, actually. It's not that I don't know, it's that we don't know in the field. It's something we are testing. So there could be co-release of different neurotransmitters here. Yeah, that is, I'm not sure if two different neurotransmitters can exist in the, do we have any evidence of that? Two different neurotransmitters. Experience-based neuroplasticity can explain that. Like, I can behaviorally. Yeah, but physiologically, like GABA and glutamate might not exist, and it's kind of confusing for the synapse to have both at the same. So you'll have two different inputs onto the same synapse. It is possible that one spine can have both GABA receptor and a glutamate receptor, but presynaptically, you might not have that. So presynaptically, the inputs will be still segregated, but postsynaptically, you can have dually innovated. That's a neuromodulator. That's exactly what we discussed. right? So neuromodulator is is you can have neuromodulator synaptic vesicles that can coexist with your neurotransmitter. You can have glutamate and dopamine. You can have serotonin and GABA, but having acetylcholine and glutamate, I'm not sure. This one?
One of the synapse, I mean one of the atoms instead of the other. So there could be three combinations. One
You increase the probability, yeah.
One of the one. So that could mean different things very, very, very.
Absolutely. You can have that. Although I feel like what this is doing is with the same presynaptic partner, you just you are increasing the computation. So non-linearity in how they can get activated, right? So absolutely, all of that is true. Mhm. No, that we can quantify. But what we don't know is in what physiological context you will have an increase in the spine volume versus you'll have an increase in the spine number. All I know from evidence, again correlational, is that in a lot more dramatic event, for example, if you look into the number of spines over age, if you take an 80-year-old man's hippocampal neuron versus a 25-year-old, you'll have a lot less synapses. So I know that it really the number of the synaptic density changes in a lot more dramatic event. For example, if you undergo like chronic stress or if you have a very traumatic event, right, then you can see changes in the spine density. Whereas in normal physiological learning, for example, you're learning for an exam, that might not lead to changes in the density of the synapses. It might be more of the spine volume changes. Absolutely. So absolutely. So like if you have these very dramatic events. All right, but great points that we can keep discussing. But let me move forward on understanding the mechanisms. You saw how we induce this high frequency stimulation as a way to show synaptic changes and increasing in the synaptic strength. Like that, we have really developed a lot of different ways of inducing plasticity for different time scales. Okay, you can have short-term plasticity that can just last for a few seconds to minutes, and the mechanisms of that is different. It would all involve one of those 3 or 4 things of the synapse. You can have long-term plasticity or you can have homeostatic plasticity. Okay? Now, short-term plasticity examples are post-radical potentiation. I'll discuss a bit about that. And then sensitization and habituation. Again, these are all short-term. They last for a few minutes to seconds. LTP is what we discussed. This high-frequency stimulation is one of the ways of inducing long-term potentiation. And if you give that in a low frequency, you induce something called as long-term depression. So these synaptic changes are bidirectional. Then some new, so STDP is a spike timing dependent plasticity where you have the presynaptic neuron firing before the post. So I'll talk a bit about that. And these can last for many hours, at least in our recording conditions. And so I'll talk about the differences between BTSP and STDP. And then homeostatic plasticity is again something which is a very cool concept, but we have not been able to study that. So far, we study this as a very different plasticity of any large network level changes can really cause some dramatic changes in the cell or the neurons that are experiencing these network level changes. So for example, your cell is there and you remove all action potential dependent release from that. So you apply something called as a TTX, sodium channel blocker, for like 48 hours. What you'll see is to kind of maintain its electrical excitability, it just increases the number of synapses so the increase in the number of synapses, in the sense you'll have a lot more spines. So that's called as a synaptic upscaling. And same way, if you remove inhibition for like 48 hours, which is again a very dramatic thing, you'll just do something called as synaptic downscaling. Now again, what homeostatic means is that it is a compensation to something dramatic that's happening within the neuron. So we do something which is very crazy dramatic in neuroscience, but even this short-term plasticity is an event which needs some compensation, right? Even long-term plasticity is an event that needs some compensation. You are changing the strength of some synapses, so you need to have some checkpoint such that the other synapses don't grow and increase the excitability of the cell after a certain limit. Otherwise, cell is gonna go into electrical cytotoxicity, right? So you need to have some homeostasis happening at all these different types of plasticity as well. Something that as neuroscientists we have not explored a lot. Nonetheless, this is a delayed effect and this can last much longer. All right. So we'll just quickly go, let's see. We'll just quickly go into the short-term plasticity. This is generally believed to be presynaptic. We learned about probability of release, you guys remember? So this is telling you what is the probability of the synaptic vesicle release when an action potential comes down. You have you have some synapses with low probability. The example of that is hippocampal CA3-CA1 synapse. When you give low frequency stimulation or you give like repeated stimulation at low frequency, you start to see something called as a facilitation. Okay? And now you guys can think of why this could be the case. So here you are somehow increasing the probability of release. And that's why you see a bigger EPSP or EPSC in this case. Right? What could be the reason? Yeah, absolutely. So there's a residual calcium that remains in the presynaptic terminal, and as you keep giving it again, the calcium gets added up. So the probability increases. On the other hand, if you start from the high presynaptic or high probability of vesicular release, you start to see something called as a depression when you give this low frequency. Why is that? Yeah, so depletion of, even though you have high calcium in the first one, you start to deplete the readily releasable pools or the primed vesicles. And so that's why it will go into depression with this. So these are some of the short-term plasticity changes. Suddenly, this will remain only for like maybe few seconds. So after like a 1 second or two, again you start, it will start from the baseline, right? So this is also a form of plasticity. And okay, so this I should also tell that this is also, I think, one of the reasons why we see something called as a post-tetanic potentiation. So you record baseline, and I showed you the LTP. It looks like this, right? So you induce 100 hertz LTP. This is EPSP, EPSC amplitude, right? So 0, 100, 300, 200, and this is time. You guys remember this? So you have this initial increase and then it decreases and then stabilizes, right? So this initial increase, which I termed it as induction, involves something called as a post-tetanic potentiation. In this, what happens? You have a presynaptic component getting added as well. So when you give this high frequency stimulation, there is a huge accumulation of calcium into the presynaptic terminals, and that leads to an increased release in the probability for like maybe few minutes. And then it comes back down to baseline, where this part is mostly postsynaptic, at least for the CA3-CA1 neurons. But this is due to an increase in the probability. So this is also a short-term plasticity that get added on top of the LTP. Absolutely. So that's why you initially see facilitation, but it depletes also very quickly. But it's somewhere in between. All right. So I want to describe these two plasticity mechanisms. The reason is because we study this in our lab. There is something called as a spike timing dependent plasticity, where this is very Hebbian in the sense it says only when a presynaptic cell fires before a postsynaptic depolarization. Pre comes before post. Only then you will see potentiation. So you're recording baseline EPSCs. Presynaptic stimulation leads to an EPSC. And then you have a postsynaptic depolarization that leads to firing of the postsynaptic. When you pair them together, you see potentiation. But this needs to come before this. And this pairing can only happen between 0 to 40 milliseconds. Okay? Why is that? Molecularly? Because we might not need to reach there. That is because NMDA receptors need to get activated. Right? So both glutamate release and postsynaptic depolarization leading to magnesium block has to happen almost together. Between 0 to 40 milliseconds is the time when the coincident detection can happen. And this NMDA receptors act as coincident detectors and activates bunch of signaling cascades. And that leads to potentiation. But interestingly, we observe that when the postsynaptic depolarization happens before the presynaptic inputs come in, such that you have the firing before the EPS. Okay? That is in your control. So you're recording from the cell and you're stimulating the presynaptic axon. So instead of stimulating the presynaptic events before the postsynaptic, so depolarization you can give by giving an artificial current injection. So either you give presynaptic first and then postsynaptic depolarization, or you reverse it. You give postsynaptic depolarization first and then presynaptic inputs. But in this case, you see a decrease in the EPSC amplitude. Exactly. So this is, so this is an increase 0.4 to 0.6 nanoampere. This is almost a decrease, right? And this is what the summary graph look like for spike timing dependent plasticity. And this has been in the textbook for 20 years. And we thought that this is actually what's happening when we learn something, right? So when you have so the depolarization acts as an instruction signal, and then the inputs acts as a information that's coming into that cell, right? So when both of them are happening around the same time, depending on what happens, you either see an increase in the synaptic strength or learning happens, or you see some kind of forgetting or extinction happens. And we based a lot of our behavioral paradigms based on this spike timing. Okay? Although there are some problems here. One of the problems is this pairing is happening over milliseconds, right? This is not how learning happens. The learning is happening at least over seconds or minutes, if not milliseconds. And so a lot of computational scientists kind of weird it out about like, how do we really use this as a model? So they try to change this temporal window in many different ways. They either add like a neuromodulatory component that can increase the window, or they add a lot of different components, which I'll not go into detail. But still, this kind of felt a bit incomplete to explain the learning. And then came this new plasticity in 2017, 2018, which kind of changed the game. And this is something that we are studying in the lab, which is a lot more behaviorally relevant. And it is non-Hebbian in nature. And I'll explain you why it's non-Hebbian. Here, again, in the hippocampal slice, looking at CA3, CA1 synapses, we have the presynaptic inputs that get activated. So we just give 10 non-Hebbian. That's exactly what I'm explaining. So you understood the Hebbian form, where the Hebbian is more of a potentiation only in one direction, pre and post and pre is depression. But the reason why we call it non-Hebbian here is here what we do is we have the presynaptic inputs first and then the postsynaptic depolarization that leads to potentiation. So this depolarization is in the form of something called as a plateau potential, or these large depolarization events. But we are pairing them over several hundred milliseconds or almost seconds. Okay? And that leads to an increase in the EPSP amplitude. But in the protocol, when we have the postsynaptic depolarization, before the presynaptic inputs, even that showed increase in the EPSP amplitude. So this is different from what we saw before. And it is these large depolarization events that they thought was able to do this kind of a non-Hebbian integration of inputs coming in. So this is a plasticity that we study in the context of spatial navigation, or how the place cells or cells that encode location-specific information, how they get induced. And they found this kind of behavioral timescale plasticity. And if you look at the summary of that, regardless of where you give the postsynaptic depolarization, either before or after the presynaptic inputs, you will see potentiation, and it will be higher than 1. And you can pair up to 4 seconds where the presynaptic can come after the postsynaptic or before the postsynaptic depolarization. So this is the non-Hebbian part, where in the other case, you have depression and potentiation depending on the order of when you're giving. But here, it doesn't matter the order, you can get potentiation in all of them. Okay? And so I'll skip this due to the interest of time because I want to get into the mechanism of how this plasticity actually now happens. Like now, let's start visualizing about what really is happening. So I'll skip this as well. And this, I wish we had like several classes of this because there are a lot of cool features, but I'll skip this. Let's go into the molecular mechanism of molecular mechanism. So yeah, so let's go into this, and then if time permits, we'll go back into some of the cool observations that people have made to study plasticity. Now, one of the cool things happen during normal synaptic transmission. All you have is activation of voltage-gated calcium channels. Calcium goes in, some proteins involved in synaptic vesicles, chemical release, and you have ion influx. But when patterns of electrical activity or neuromodulators try to change the properties of synapses, there is a beautiful electrical to biochemical signal transduction that happens. And depending on the type of inputs that come in, you activate a very complex myriad of biochemical signaling. Okay, and just want to show you how much scientists have now identified that by studying these different types of synaptic plasticity mechanisms. But you can see, you can appreciate how both at the presynaptic and at the postsynaptic, this is not, of course, not the exhaustive list, but you have so many proteins that can really do one of those three things that we discussed in the beginning, which is vesicle number or more spine volume or more number of receptors or more spines. But why? So one of the questions in the lab that we ask is why? Why do we need these thousands of proteins to do one of these 3-4 things? Can't we have just like 4 proteins for these 4 changes in a bidirectional manner? And this is something that we are trying to test, like why do we need it? And one of the hypotheses is this. We think that this is another way of visualizing it. So electrical stimulus is 1 to 2 milliseconds. Whatever learning stimulus that needs to come for making the changes, it's 1 to 2 milliseconds. And that activates different types of signaling cascades, right? Some signaling cascades can stay for 5 to 10 seconds. These are kind of the secondary messengers. Can you guys think of some examples of secondary messengers? Cyclic AMP comes first. Protein kinase is downstream of cyclic AMP. It's not a secondary messenger. It's a kinase that gets activated. IP3 DAG is, IP3 again is one of those. Calcium, exactly. So one of the main secondary messengers for plasticity is calcium, which is an NMDA receptor dependent, right? So you can have some secondary messengers that can last for a few seconds. Then you can have activation of downstream signaling cascades like PKA, like protein kinase, like kinases or phosphatases that can last for a few minutes. And then you can have a lot more gene expression changes or structural changes in the synapse that can last for hours to even years, right? And depending on the type of signaling cascade, you can have either information stored for few seconds, few minutes, or for long term. And so we think that the short term and the long term memories are really dependent on the type of signaling cascades that get activated. Now, how do we study them? How do we really understand these signaling cascades in the context of learning? We need a lot of functional tools, right? Now, over years, we have studied these protein signaling cascades by different ways. And there's an evolution of that. So in 1950s, we use pharmacology to specifically block drugs. Either we can give drugs to study this plasticity, we can give the drugs in intraperitoneally or intravenously, and then see the effect. Or in slices, we can apply it in the bath. Or when we are patching the cell, we can put it in the internal solution, but we can block these specific protein activities and we can check the function of proteins. Then came, once we started to understand the genetic architecture of the neurons, we started deleting some of these proteins and see if we can have a knockout. How would it impair different forms of plasticity? And this was a little bit more cleaner than this, because pharmacology always would have some off-target effect. It's not usually very specific to a particular kinase, right? So this was in 1990s. We still use both of these techniques, but there are some limitations to this, where here we're only getting a qualitative information of when does whether a protein is required or not required, right? That's it. That's all information you can get. But now we have started visualizing protein activity. And this is something that I'll talk about. This is something that we are trying to do in the lab, is to now quantify when does a protein get activated, for how long it gets activated, and where does it get activated in the neuron. Is it throughout the neuron, or is it in some specific compartments? And that can really give you a better idea on the function of these proteins and the rules of how these proteins are getting activated. See, so we already discussed this, where we now know. So this is an example of a pharmacology where you have a control, and you see the same LTP. This is in CA3-CA1 synapses. But when you apply a drug, APV, this is an NMDA receptor antagonist, they found that the plasticity is blocked. And this is kind of, again, textbook information, where they found that NMDA receptors are critical for LTP induction. And they have now found, not just in hippocampus, but in different brain regions, NMDA receptor dependent plasticity is actually predominant. But in some of them, even though NMDA is important, it's not the only source of calcium. There are these entorhinal cortex. So there are these direct perforant pathway where entorhinal cortex makes synapses to CA1. In those experiments, when they block NMDA receptors, they did not completely block the potentiation. Why do I say it did not completely block? In this graph, so this is a control in green, and APV is here. So why do I say it's not completely blocked? I was like gone. Simple, no? As compared to baseline, this is still above the baseline. So it's a partial block. Only when I block both NMDA receptors and an L-type calcium channel, which is also a source of calcium, then I see a complete block of plasticity. So these are some examples of using pharmacology to really try to tease out the different calcium sources or the different proteins that are important for LTP. And then again, we can go more detail into like downstream of NMDA receptors, what gets activated. For example, in this one, they say that the dentate gyrus, which makes mossy fibers, the axons are called mossy fibers, to CA3, they are not NMDA dependent. So they are dependent on pKa, but they don't really require intracellular calcium. So in this, the secondary messenger is different, right? There's activation of cyclic AMP. So only when they block pKa, they see decrease in the plasticity, but not with APV. And so even within hippocampus, you can see that the mechanisms of inducing plasticity are diverse, right? All right, I thought of explaining this NMDA dependent signaling. So let's just quickly go into what happens and how it basically leads to the changes in the synaptic strength, what we kind of know about it. Again, this is something that I'll just explain the basics. So you have a synapse, and then you have the AMPA receptors. I have two colors, so yeah. And then you have NMDA receptors, okay? So you have more AMPA receptors. So when NMDA receptors get activated, there is influx of calcium. So that acts as a secondary messenger. When does NMDA get activated? Coincident detection, pre and post, so something important is happening. And then calcium leads to what? It binds to something called as calmodulin. And it activates something called as a CAM-K2. Okay? calcium calmodulin complex binding protein kinase. I don't know. Calcium calmodulin. What is it? Activation kinase, complex activation kinase 2. Something. But it's basically the calcium calmodulin activates this CAMK2 protein. And this CAMK2 protein, again, is considered as one of those kinases that is very important for plasticity. Now I'll tell you one thing cool. So, and then what happens is what does CAMK2 do? CAMK2 can do many different things. So it can basically increase the number of AMPA receptors by phosphorylating some of these AMPA receptors that exist outside, and then it laterally moves them into the synapse. There's a lateral movement inside the synapse, and it immobilizes them. So CAMK2 immobilizes these movement of AMPA receptors into the synapse. Okay? So translocation में CAMK2 can play a role. CAMK2 can also phosphorylate some subunits of AMPA receptors to increase the conductance. So even if the number of receptors stay the same, there can be overall more conductance of ions. So they'll either be open for much longer, their open probability of receptors increases. And so the CAMK2 itself has a lot of different function. It can also maintain the synapse in a bigger spine volume so that more receptors can be activated. And then the calcium can also then activate something called as a calcium dependent, what is it? What is before cyclic AMP? So it can also act, what's the, so how does cyclic AMP get activated? Adenylyl cyclase. So there's a calcium dependent adenylyl cyclase that gets activated, adenylyl cyclase 7, that can activate cyclic AMP, and you can also have activation of pKa. And pKa can also participate in all of these other functions. So pKa is another protein which is very important that people have identified that can mostly change the properties of these conductance of the receptors. Then you can have activation of CAMK2 downstream can activate something called as a SIR Rho kinases or ROC. And then you can activate MAP kinases. So MAP kinase can lead to, CREB in the nucleus, can do transcription or gene expression changes. ROC can do something called as a polymerization of actin cytoskeleton. So this is the one that plays role in the structural plasticity. Okay? So there are a lot of these signaling cascades that are NMDA dependent. So just by activating NMDA, you can activate some of these. Now, what if I tell you that this is a very beautiful, clear-cut understanding of potentiation, but a lot of these players, or at least NMDA, is also important for LTD in the same synapse. Can you believe that? NMDA is also important for LTD. It's important for LTP. We also saw that. But if you block NMDA, it also blocks LTD. How is that? Isn't that crazy? Like NMDA receptors are actually important for both LTP and LTD. What? It activates NMDA? How does that? Because you're right, you could have asked this question that pre or post me LTP kyun hota hai? Post and pre, if you do, you are still activating NMDA, but why do you get depression? And we know molecularly it's depression, isn't it? So it is true. I mean, you guys don't know this concept, so I'm not expecting that answer. So NMDA receptors, when you have during LTP, depending on the calcium profile, so if it's high amplitude and it goes down very quickly, you have an LTP-like signaling that gets activated. If it is low amplitude and low decay like this is more of an LTD. And so even though the NMDAs are the same, but quantifying the secondary messenger signaling becomes important, right? And this is where you can start to appreciate why quantifying is important. Just telling whether something is important or not important really doesn't tell you about why, but this will tell me. So that's when we started doing calcium imaging in the synapses, we started to see this kind of profile, which is different from LTP and LTD. Okay. Okay. So similarly, I said that now we can quantify different proteins underlying synaptic plasticity, and the way we quantify that is by developing sensors that can detect different specific protein activities. And this is something that we are doing in the lab, where we make PRET sensors, where you have a donor fluorophore that is bound to some part of or some subunit of protein, and you have an acceptor fluorophore. Now, if there is a conformational change, the donor and the acceptor fluorophore will either come close together or they go far apart, and that can lead to differences in the FRET efficiency. So either there is low FRET in the beginning, after activation it increases, or vice versa. And that we can measure. So we can measure the changes of the FRET either by measuring intensity or by doing something called as the fluorescence lifetime imaging that I'm not going to go into detail. We can see when and where these kinases get activated. Okay. So this is a real data that I'll show, where we have now developed a sensor for this protein called as CAMK2 that you know it's important, right? And I'll show you that upon giving a learning stimulus, there will be an increase in the spine volume, which is telling me that there is an increase in the strength, right? But before the spine volume increases, you will see the activation of CAMK2. You'll see that the color of this dendrite or this activity changes, which is represented in the form of a heat map. Okay? So you want to see? Let's see the video. So upon stimulus, see the activation happened, and then there is an increase in the spine volume. Now this is in hippocampal synapses. We are just looking at one dendritic shaft. So what does this tell you? This tells you that CAMK2 activation happens before the potentiation actually happens, right? So CAMK2 is actually important for causing that change in the spine volume. Second thing, it is restricted to the synapse. It's not really spreading to the dendrites. You saw that, right? Let's see that again. And it's very acute. It's really stimulation locked. It activates as soon as you start giving the stimulation. It is restricted to the synapse. It's not really spreading to the dendrites. So you see how we can quantify this a lot better than just blocking CAMK2. Isn't that's kind of boring, and it will not give us this information. Whereas some other kinases, if you see, now this is like a bigger chunk of the dendrite, because we want to show you, this is an example of Rho kinase, Rho A. I told you here, right? It's important for the structural plasticity. You see this kinase is a lot more spreading. Right? Cannot play. All right, I'll share this with you guys later. But what you'll see here in this spine, if you give the induction, the spreading will be throughout the dendrite. And that, we can start to quantify now the activation profiles of some of these proteins and understand their functions, right? Maybe it is possible that CAMK2 is only playing a role in providing synapse specificity during plasticity, but Rho A is kind of participating in priming other synapses that could also play a role in the behavior or clustering in these synapses. And so like that, we have developed a lot of these different kinases. Some of them can get activated within seconds of giving any kind of learning stimulus. And they could be synapse specific. So all the greens are synapse specific and the reds are spreading signal. And some of them are activated in a lot more delayed manner. And you can start to now map out the different functions of these kinases depending on their activation profile. So we have some of these sensors, and we are developing some new sensors for other proteins that could also play a role in different relevant plasticity mechanisms. Cool. I think that's it. I think we can just end there, right? Any questions?